For our final project, we created a bioinformatics tool that is used to compare genetic sequences. We enter a sequence and look in the database for similar sequences. We focus specifically on sequences found in microbes. We originally contacted the developers of BioCaml. BioCaml aims to be the standard library for the biology domain. It has input parsers, algorithms, data structures, and hooks to link to public repositories. We decided, however, to build our own standalone tool with hopes of linking later on to BioCaml. Our DNA and protein sequence alignment tool has three parts. We have a parser that takes in a file and turns it into our data structure. We have algorithms that run for comparison, and we have output to HTML that can be read into a browser. I will talk about the parser and the output and hand the rest over to my partner to discuss the algorithms and the tool demo. When running the tool, we parse the command line to get the file name where the sequence is held. We open a channel, we read each line one at a time and cons onto a string list. Then we parse that string list and convert it into our internal data type, which is basically a sequence and a comment. We have some additional functionality in our parser. We can parse multi-files, and we also have debug features where we can print out what we read in and also what we stored for debugging purposes. For our output, we open a channel and create a new file called output.html. Inside our algorithms, when they're finished, they print out the sequence to this file. We open a channel that now appends to this file, and we print out the score, the query sequence, the test, and the comments around HTML tags. The same output sent to the screen. Now we'll get to an interesting part of our tool. We'll discuss the internal algorithms we developed as well as a tool demo.
So we used three different types of sequence alignment algorithms, one for global alignment, one for local alignment, and one for repeat alignment.
The global alignment algorithm was used to align two entire sequences and determine their similarity. The local alignment algorithm was used to find the most likely occurrence of a sequence within a larger sequence, and the repeats algorithm was used to find repeats of a sequence within a larger sequence. So our goal was to assess the similarity between sequences and to determine whether they may have arisen from the same sequence. The challenge, though, is that over time, mutations can render sequences less similar, making alignment hard. Because of this, the algorithm needs to use a score for every possible match or mismatch, and it also references a substitution matrix to keep track of these scores and determine scoring for a given match or mismatch between subunits. This was the DNA scoring matrix we used, which is fairly simple. You can see a match is 2 points and a mismatch is a penalty of -1. And this is our more complicated amino acid scoring matrix. So each algorithm stores the scores for each potential subunit matchup in a second matrix, which it populates as it traverses the two sequences. And so you can see a basic idea of what the second matrix looks like. So each box gets filled with a score and a pointer to another box. And the score in each box of the matrix basically inherits part of its score from another box, usually the box on the left, the box above left, or the box directly above. The best case is if it inherits something from the box to the above left. That means that this is probably a match. If it's inheriting something from the top box or the left box, that means there isn't a counterpart to the current subunit in one of the sequences. So each algorithm keeps track of its path as well by keeping a path value in each block. And for this, we used a tuple in our two-dimensional array, and the tuple stored a score and it also scored a path. Once the final score has been determined, the algorithm uses the path values to trace back the path to its start to determine which subunits were aligned with which. And so you can see kind of a sample traceback path here. And you can also see here, was an actual set of sequences we used. These are protein sequences, and you can see that the traceback path starts in the bottom right-hand corner and goes all the way to the upper left-hand corner. And using the path, the algorithm then computes the sequence alignment. And you can see basically the dashes correspond to upward or sideways arrows, and where letters are filled in, those correspond to the diagonal arrows, which again are kind of the best case matches. So here is a quick demo of BioStat. I'm just going to enter this into the prompt. The starting command, you'll notice it takes several arguments, the type of sequence, here I have protein, the type of algorithm, here I have global, and the output file. So we're going to go right ahead. And we're going to enter the lowest possible sequence score. I'm going to just have this as 0 because we don't need to worry about that right now. We're going to have the maximum number of hits. I'm going to set that to 10. And here it's asking me to input a sequence or file path. I'm going to select a prepared sequence called protein query.seq. And you can see that it outputs a sequence alignment and a score that is consistent with our source material that we used as a reference for our project. And here, you can see that the resulting output file does in fact open in a browser. Because of its straightforward prompts and its flexibility across DNA, RNA, and protein sequences, we do feel this is a potentially useful tool for biologists who want to examine evolutionary relationships between specific sets of organisms. So that is an explanation of the algorithms we used in our sequence alignment tool, and we hope you enjoyed it.